pick things back up here and we'll continue on with the cell. So inside the cell, in addition to all of the organelles that we also potentially have a so-called skeleton of the cell. So we have the cytoskeleton. This is essentially going to be a series of proteins that allow for us to maintain the shape, to protect the cell, to allow for some movement in the cell, essentially giving the cell its overall structure that we see. And so we have things like microfilaments, we have intermediate filaments, and we have microtubules, all going from smallest to largest and having some changes that occur within each one to alter those proteins that we find inside. So microtubules are essentially a long tube that we have for them. They tend to be found in places like cells that have either cilia or flagella, as they serve as part of the structure of each of those flagella and cilia. And so they're surrounding them, creating part of the base of them to help with movement, as well as with centrosomes for help with movement during reproduction of the cells themselves. Microfilaments, these are structural proteins, and so within them, they oftentimes provide for strength of the cell in terms of maintaining its overall shape, as well as providing for a means for uh, motor attachment of special motor proteins that allows them to move along the surface of the cell potentially, or to move organelles within the cell, allowing for us to kind of shuttle things along. So back when we talked about transcytosis, this would have been part of what they are allowing to occur. And then we have our intermediate filaments. And so intermediate filaments are the ones that are going to essentially keep cells from getting stretched out. They have a high tensile strength to them. And so when we have cells that have intermediate filaments in them, it allows for them to essentially be pulled on, twisted on, things like that, and the cells themselves aren't going to become damaged from. Connecting to those actin molecules that we have for movement, we have special motor molecules. And so these motor molecules are going to utilize ATP as energy, and that energy is going to come along and allow us to move the cells or move organelles or move parts of the cell and allow for that attachment site for our motor protein. So here we have our microtubule that has the actin on it, and then we have our motor protein, and that motor protein then is essentially kind of able to walk that particular organelle along the cell. In other cells, those motor proteins and the um, microtubules are going to allow for us to move the cell in a way. And so allow them to kind of alter their shape or alter their movement. So in things like white blood cells, we have the ability to kind of move them along through the tissues themselves. In cells that divide, we have centrioles. And so centrioles are a special protein inside the cells themselves that take microtubules in the form of triplets. So we take 3 of them at a time, and we're going to combine 9 of those triplets together. And we're going to make something that, to me, always kind of reminds me of something like a gear. And so it kind of looks like a gear when you look at it from the side. And this is going to attach to microtubules to allow for that movement of things like cilia and flagella. So they're going to attach at the base of those structures, and those microtubules are going to travel up through into them and allow for that movement of the cell itself in there. And also found as a vital component for mobilizing the DNA when we divide our cells. So here we have it with cilia. And so here we have our triplets once again. And so we have those triplets at the base of itself. And then the microtubules are extending up through the cilia. And as that base, in a sense, kind of spins around and around and around and around, that's going to allow the cilia to stand up straight and then bend down. And so When it stands up straight, it's in what's called its recovery stroke. And so it's going to stand up rigid. And then it's going to forcefully push down. And what that does is it makes the mucus that may be on the surface of those cells travel along. And so the mucus is going to be pushed along the surface of the cells themselves. And so this is going to be found in places like the respiratory tract, where we want the mucus to get up and out of the deeper areas of the respiratory tract. The nucleus is the place within the cells where we have all of our genetic material for protein production and cell reproduction. And so this provides for that so-called genetic library that we have within cells, provides for all of the components that each cell is going to need to have in order to make all the proteins that it has. Every single cell within a body contains the same genetic library that's there, just that each individual cell does not necessarily know how to read the entire library. So it doesn't know how to make all of the different proteins that are inside, which is part of the reason why when we have something like damaged tissue, we can't just replace it with cells that we grew or something like that of another type. And so we can't just take muscle cells and make skin cells, or take skin cells and make muscle cells out of them, because those cells, even if they are stem cells, they don't know how to make the other kind of tissue because they can't read the other part of DNA. So the majority of the time, when the cell is not dividing, all of the nuclear material is condensed into chromatin. So when we look at that cell under the microscope, it's going to look like basically just a nice clear nucleus. It's going to have stain to it, but it's not going to have any kind of visible structures that we can see in there. Sometimes cells, we can see the nucleolus, depending upon how big the nucleus itself is. And so we may be able to see the nucleolus there. The nucleus is similar to that of the cell itself in that it has a membrane. And so it has, rather than a plasma membrane, it has a nuclear envelope. And so that nuclear envelope essentially surrounds all of the nuclear material inside, all of our chromosomes inside there. And it actually has two layers to it. So it has an outer membrane and it has an inner membrane. And that's going to regulate what is allowed to come in and what's not. And so the nuclear envelope, rather than being semi-permeable, is selectively permeable. It only allows for certain substances to get in and out of that nucleus. One of the other big structures that we have inside the nucleus, as I mentioned before, is the nucleolus. And so the nucleolus is the site for ribosome production. And so we're going to make ribosomes inside the nucleus. Those ribosomes, remember, are going to go out and allow for the production of proteins. So one of the main jobs of the cell is to produce proteins, and another main job of the cell, as far as the nucleus is concerned, is for cell reproduction. And so the cell itself can be divided into two main phases. We have interphase and we have mitosis. And so we have our interphase, which is divided up into subsections as well. And so we'll look at each of those. We have a G1, an S phase, a G2, and then there is also another G0 that we'll talk about. And then mitosis is divided up into phases as well. And so we have prophase, metaphase, anaphase, telophase, and cytokinesis, going from getting ready to divide the cell all the way up through cell division. And we have now two separate cells at that point. So for the cell cycle, the interphase that we have, generally with most cells, it goes through G1, and then it goes into an S phase, and then it goes into G2. And so it's going to move through those three phases. And then once it's gone through G2, it's then going to go into mitosis. So we're going to go through cell division. Both of the G phases, our gap phase 1 or our growth phases that we have there, are all about growth of the cell. So in G1, we've just made the cell, so it's going to need to grow itself. It's going to need to make more organelles. It's going to increase in size. G2 is increasing the cell in size for cell division. So whereas in G1, it's basically just getting larger in order to be able to function and do its job in a sense. G2 is all about making the cell much larger than it was so that it can support essentially two cells once cell division has occurred. But before G2 can occur, we have to go through the S phase. And the S phase is our synthetic phase. This is where DNA is becoming replicated. We're making a second copy of the DNA. If a cell isn't going to divide anymore, that cell may oftentimes become stuck in a sense in G0. And so G0 is a situation where the cell is no longer going to divide. So this would be things like muscle cells. And so skeletal muscle, cardiac muscle does not divide ever again once we have it. And so what we have in a sense when we're born is just about what we have when we will be an adult for the most part. Other things that don't divide, things like nervous cells, and so in general those are thought to be non-dividing cells as well, so they're going to be stuck in G0 also. But when that cell does need to divide, or when that cell needs to manufacture proteins, we need to be able to take the information from the DNA and copy it. And so when it comes to making proteins, this process itself is going to use, when we make proteins, we're going to use RNA. And so RNA is going to be our substance that is going to code or have the code on it for the recipe. And so in a sense, we kind of have the nucleus. And inside the nucleus, we have a book. And so that's my big book there. And inside that book, we have then all of our little recipes. But we can't take that book out of the nucleus. So what we're going to have to do is write down the recipe. And so first we're going to do what's called transcription. We're going to write down the recipe in a sense on a note, and that note is going to be on mRNA. And so mRNA is going to be where we're going to write that down. So in order to do that, one of the things that we have to think about in terms of DNA, I'll just skip over to the picture here, is that DNA is a double helix. And so since it's a double helix, that means that when we go to do and make proteins, we need to pull that double helix apart. When we go to make a new set of DNA for cell replication, we're going to pull that DNA apart permanently. And so for RNA polymerase, we're going to pull the nuclear material apart temporarily. But for cell division, DNA polymerase is going to permanently detach and unwind our DNA so that we can make two separate copies of it. When we do that replication of the DNA, it's said to be semi-conservative. And what that means is that if you look here, we have two pieces of the DNA, two forks there, that allow us to split it. And so each one of those, in a sense, is the old DNA. This is what we call the template. And so the template is going to go onto the new DNA along with a completely brand new strand. And so we've got new strand, we've got new strand. And so each one has, in a sense, a portion that is the old strand, two strands there, and then we've got a portion that is the brand new strand. And so both of them contain half of the material is old, half of the material is new. And so this is this semi-conservative replication. We're conserving part of our DNA going into the new cell. So that new cell always has essentially half of the information came from the original source, being the cell that it was divided from. So for cell division to occur, we need this for normal tissue repair. We need it for normal tissue growth of the body itself. And then we also need to be able to do it simply for the replacement of lost cells as well. And so this gets divided up into two portions of the mitotic phase. And so we have mitosis, which is what we call the nuclear division portion. And then we have cytokinesis, which is where we actually divide the two cells themselves up. And so we're going to divide up all of that, not only the nuclear material, but then all of the organelles between the two cells as well. So for mitosis to occur, we have some phases, and each one of them tends to have also some intermediate stages as well. And so we have things like pro-metaphase and pro-anaphase and things like that we can see in other books and so on. But each one has kind of a unique set of events that is occurring within each one. And so for prophase, essentially the big event that is occurring from within prophase is that the chromatin condenses into chromosomes. And so we go from essentially having a nucleus where there's not really anything visible inside to where that nucleus almost looks like it has worms in it. And so later in prophase, not only do we have those condensed chromosomes, but we also have the nuclear envelope disintegrating. And so the nuclear envelope is going to completely fall apart as well. Those chromosomes end up attaching to our centrioles, and they end up getting pushed towards the equator. And so the center of our chromosomes are called the centromere, and those centromeres line up all right on the equator of the cell. And so they're nice and lined up at the equator, and essentially half of the chromosome is going to go this way, half of the chromosome is going to go this way when we start to do cell division itself. And so during anaphase, this is where this is starting to occur. And so we're starting to pull those nuclear material to either side of the cell towards the poles. And so those motor proteins are pulling on all of those chromosomes, and they're moving them across to either pole of the cell. And then that new developed set of cells is going to start to grow up the other side. So we'll see that in between there as well. The last phase in terms of mitosis is telophase. And so with telophase, we now have all of the nuclear material out towards the poles. We can start to rebuild, kind of the lighter purple color here is the nuclear envelope. And so those can start to reform inside the cell itself once it's there. And then in the center of the cell, what used to be the equator of the cell, we start to see little vesicles appearing, which are going to be the reformation of the plasma membrane on the outside of the cell. And so this is going to start to invaginate. It's going to start to pinch in on either side, and eventually it completely closes in, and we then have two completely separate cells. And so those completely complete the cell division process itself. And so here we can see on an microscopic image what's going on during the different phases themselves. And so here we have kind of early prophase. We've still got a nuclear envelope present. Here we have metaphase. And so we have metaphase there. All of the chromosomes are along the middle, so kind of meta-middle. And then we're going to go through anaphase. And so anaphase, we're pulling that nuclear material to either pole. And then we have telophase. And so here we're starting to get the reformation of the nuclear envelope. And then eventually cytokinesis, we're cleaving the two cells in half. And so now we start out or end up with two brand new cells that are identical to one another. So they are completely looking like the parent cell that they came from, and the two daughter cells there are perfect accompaniments of what we started with. And so they're going to have the exact same DNA, they're going to have the exact same function, and so on within the cell. We don't want cell division to occur kind of, in a sense, whenever a cell wants to. And so we have to have some processes in play that are going to restrict cell division. And so one of those is just simply what we call surface to volume ratios. And so essentially, is the cell big enough? And so does the cell contain enough material in order to make two copies in the end, two functioning cells at the end of the process? We can control cell division through things like chemicals. And so our hormones can allow for this to occur. And so we get different hormones that can either speed it up or slow it down. Depending upon where the organ itself is, we can use contact inhibition. This is going to limit the size of the cell itself. And so essentially, if the cell is too crowded, it's not going to make more of itself. This is similar to kind of like if you were getting into an elevator and the elevator doors open up and you look inside and the entire elevator is completely full. Everybody's right on top of one another. You don't want to touch them, so you have contact inhibition. You decide to take the next elevator that comes along. Well, cells work kind of the same way. They see that they're kind of bunched together and squished together, so they're not going to divide on there. And then we have cyclins and cyclin-dependent kinases. Again, chemical processes that are going to allow us to control cell division. The other side of what the nuclear material is used for is protein synthesis. And so protein synthesis is kind of the big thing that most cells are doing all the time. And so rather than dividing all the time, they're making proteins. They're making the things that the cells and the rest of the body needs in order to function. And so in order to do this, we're going to take that DNA, where we have our double strand of DNA, and we're going to have to take that double strand, and I'm just going to draw the one side here. And we're going to take that double strand and we're going to separate it. And so we're going to split it apart momentarily in order to get the copy of the genes written down. And so on the nucleus, we're going to divide this up into what we call triplets. And so we're going to take 3 nucleotides at a time and write those down. Each one of those triplets is going to code for an amino acid. And so it's going to code for a specific amino acid that we're going to need in the cell. So in order to get to the place in the cell where we make proteins, which was our ribosomes, we need to write down that copy of the recipe. And so we need to transcribe it. Transcribing it is going to allow us to make mRNA. And so this is the messenger that's going to go from the nucleus out to the ribosome. At the ribosome, we're going to have ribosomal RNA, and this is going to be a structural component of the ribosomes themselves. This is going to help with the production of our proteins. And then we're also going to have tRNA, or transfer RNA. And so this transfer RNA has on it amino acids. And so bound to the RNA is a particular amino acid. And so we started out with the DNA and we had our triplet. And then we go to our mRNA and we have what we call a codon. And then we go to our tRNA and we have what we call an anticodon. And so that's going to be the progression of information in order to get us to a protein itself. So transcription itself involves the process where we've got to take that DNA double helix and we've got to momentarily split it. And so in order to split it, we've got a few different enzymes that are going to be involved in this process. And so we have what are called histones, and those histones are going to essentially loosen up the area in terms of that double helix. And so it's going to start to kind of unwind it. And so it's going to take it from a nice tight wind to something that is more loose in a sense. And so it's going to change the winding of that DNA in that particular area. From there, we have particular segments of the DNA that are in a sense kind of marked. And these are what we call promoter regions. And so these regions are going to be read in a sense by a promoter, and this enzyme comes along, it finds the promoter, and it's going to start producing the RNA. So we're now going to unwind the RNA. We're going to temporarily do so using RNA polymerase, and we're going to essentially write down that RNA on mRNA. Once we get out into the cell, out into the cytoplasm, we're going to need to find some tRNA. And so in order for that to occur, we would have had to have made that tRNA earlier on. And so this process of writing down our DNA as a mRNA involves transcription. And so this is going to take transcription, and we're going to take a RNA. And so DNA is either adenine, thymine, guanine, or cytosine. This is what we have on DNA. For RNA, we have a little bit of a change that is going to occur. And so we still have adenine, but instead of thymine, we have uracil. And then we have guanine, and then we have cytosine. And so we still have a similar type of set of structures that are there, but instead of thymine, we change it into an adenine. So if our DNA reads so, and so this is our DNA, our RNA is going to look like this. And so where we have a, where we have an adenine, adenine normally binds to thymine, so we get a uracil. Thymine normally binds to adenine, so we get an adenine. Guanine to cytosine, cytosine to guanine. And so we have the RNA transcription has occurred now, and we have this mRNA to go on. That mRNA then leaves the DNA, leaves the nucleus, and goes out to a ribosome. And so here we just have a process where we're showing the production of transcription itself, and we're making this big long mRNA inside there, and that's going to be our product that goes out to the ribosome. Once we get to the ribosome, we have to do translation. And so translation begins with taking our big long strand of mRNA, and we're going to look for, on that mRNA, we're going to look for a codon that reads AUG. This is our start codon. And so this methionine charged initiator tRNA carries, and so this is going to be attached to a tRNA. And so that's going to be attached to that. And we're going to bring along our methionine. The tRNA is going to look just the opposite. It's going to be the complement of our AUG there. So it's going to have a U, it's going to have an A, and then it's going to have a C. And so uracil, adenine, cytosine is going to be on our tRNA. That matches up, so that brings our methionine with it, and that becomes our beginning point for our protein. So to kind of go through the different codons themselves, said that we have on mRNA, we're going to divide things up into triplets. That is there from the DNA, and then we go to codons on the mRNA, and then we go to anticodons on the tRNA. So for our codon for AUG, this codes for methionine. If we had ACU, this would then code for threonine. 3 C's in a row, we get proline, and so on. And what you'll notice here is that oftentimes, so like leucine here, we have 4 different codons that code for leucine just in that one spot. But for leucine, there's actually 6. And so we have 6 different combinations that can code for leucine. But for others, like methionine, there's only 1. And so methionine only is coded for by AUG. Serine here has 2, and so on. Tryptophan has 1. And then from there, our stop codons have 3 different combinations. And so 3 different types can code for stop. One can code only for start, and then the others oftentimes have multiple codons that can code for the individual amino acid that is being looked for at that period of time. And so what that allows for is that in our code, we can have either CUU, or if we have a little bit of a coding mistake, we can have CUC, and no matter what, either one of them is going to get us to leucine. Or AAA versus AAG, both of those are going to get us to our lysine. And so it doesn't matter which one we have, both of them will get us to the same amino acid. So if there's a small problem within the genetic code, within either transcription or translation, something went wrong, or it's the DNA itself, we can get the same amino acid, which means we get the same protein. So we have the ability to kind of allow that to occur. So the transfer itself begins on the DNA, and I'm going to skip the slide here and move to the image here to talk about it. So we start out with our triplet on DNA. So in this case, we have our TAC. T gets us an A on mRNA. A gets us a U, and C gets us a G. So our first codon there on the mRNA is AUG, which is our start codon, which means that a tRNA is going to come along with the same similar process in that A only can bind to T, or A can only bind to uracil, depending upon whether we're talking about mRNA or DNA. And then G and C can only bind to one another. So anywhere that we have a C, we're going to get a G. Anywhere that we have an A, we're going to get a U, and so on. So each one of those binds to it, and that brings along a methionine. And then we get a proline and a serine, and we keep on going along until we hit one of those 3 stop codons. And then that finishes up our protein. That stops the amino acids being added to our growing protein at this point. And as long as everything went well, we get the protein that we were looking for in the first place. That's there. Cells themselves are essentially what we start from. So we start from a single cell, and from that single cell, the DNA divides and the cell divides, and we get 2 cells, and then 4 cells, and 8 cells, and so on. And we keep on growing and growing and growing new cells. Those cells differentiate early on along the way, and so within about the first 6 days or so we have what we call stem cells. Those cells can turn into essentially anything in the body. And so our embryonic stem cells have that ability to determine where they want to go as far as the cell's production goes. And depending upon kind of the chemicals that they are receiving early on, dictates where they go afterwards. And so what kind of cells do they become? Do they become muscle cells? Do they become skin cells? Do they become a liver cell, etc.? And so that specialization starts to happen very early on in life. And as that specialization occurs, that's where we start out with our, I'm going to draw my bad book again. We start out with our whole book opened up, and we have all of the different recipes inside. But as specialization occurs, essentially some of that book becomes foreign to that particular cell, and it can only produce the proteins that it's designed to be able to make. As cells get older, part of their DNA, called telomeres, starts to become reduced in terms of their numbers. And when those telomeres start to be reduced enough in numbers, whether the cells have divided enough, or whether the cell has been damaged by other structures, by other chemicals, things like that, or even things like free radicals that we talked about, that can lead to the cell getting to the point where when it goes to divide one last time, in a sense, the cell gets to the point where that DNA codes for enzymes that are going to destroy the cell. And so we have what we call apoptosis or programmed cell death. And essentially the cell is going to kill itself before it can become a tumor cell. And so that's what's supposed to happen in cells. And so we have things like tumor suppressor genes. And those tumor suppressor genes are designed to destroy the cell and not allow it become a tumor cell that's in there. And oftentimes when somebody has cancer, it's those tumor suppressor genes that have been damaged or eliminated from within the cells themselves.